Once the DNA has been extracted out of the cell, the entire DNA I mean, now you have to make sure that you extract the gene of interest from it, because you can't do with the entire DNA. All you need is your gene of interest, or if otherwise your vector must be cut. Now what you have to do is you have to take out the gene of interest from that specific DNA. How you are going to do it? For the second step that we have in the processes is cutting of DNA to get gene of interest. Right? Now how do you cut it? This is highly repetitive at this stage, I may say it. You will use restriction endonucleases. Now when the restriction endonucleases are used, they are going to cut the DNA into small fragments. Now you have to extract those fragments. You take a test tube, right? Inside it you have the DNA as fine threads. As a result of spooling it is there. You add the restriction endonucleases. Now what you get, that there are small fragments of DNA. How to extract those fragments of DNA? For that we have the technique of gel electrophoresis. What is this? You have an idea what electrophoresis is? We have geladium or agarose gel over which we give the electric field, and according to the negative or positive charge of the particle, we see that moving through the medium. Just an overview. What gel electrophoresis is? In gel electrophoresis, the fragments which are cut as a result of restriction endonucleases being subject to the DNA, they are separated out. We have to extract out the gene of interest by using gel electrophoresis. Now what is gel electrophoresis involving? It is involving an electrophoresis plate that has agarose gel on it. Okay. For revision purpose, you can remember what agarose, where agarose comes from. That would make you revise certain parts of your knowledge from plant kingdom where we had done algae. Now electrophoresis plate has agarose gel. Such is the arrangement of electrophoresis plate. You know, DNA is negatively charged particle, right? Over here we have small wells in which the DNA, which has been subjected to restriction endonucleases, that has been cut, they are put in these wells. Okay, the DNA is added into these wells. Here we have agarose plate, okay, and to the other end we have positive charge. Now that is anode. Now the DNA when added over here, due to its affinity towards positive charge, would move towards this end, anode. It is going to move towards this end, but because we have agarose gel in between and it provides hindrance to the movement of DNA particles, what happens is we get a gradation of DNA particles being arranged over here. Larger is the size of DNA particle, smaller is the size of DNA particle, further it goes. It is quite certain. If you are if you ask a bulky person to move through the maze, the one who is fat will be making out ways like this, and he or she would be slower. But if some very swift and thin person is asked to pass through the same maze, different arrangement, then it can easily pass through it, because the cumbersome arrangement of that region will not make it difficult for the person to move through it. Larger is the size of DNA particle, the slower it moves through the agarose gel. Smaller is the size, faster it moves and reach towards the positive charge. So you get a gradual gradation where certain particles, they reach quite further and large size particles are left behind. Now this is agarose gel electrophoresis. This is the electrophoresis plate over where you see the DNA being arranged according to its size. So you get a size pattern arrangement. Those which are smallest in size, they move farthest. Those which are larger in size, they stay backwards. This is what is achieved on the gel electrophoresis plate. Now, DNA cannot be seen in visible light. White light is not capable enough to make you see DNA. So, what is done is that you use UV light. In ultraviolet light, when DNA is stained with a dye known as ethidium bromide, it glows as orange band. So, you see these small DNA fragments as orange bands arranged according to their size. Under UV light, when stained with ethidium bromide, DNA would show itself in the form of orange bands arranged according to the pattern. Then you can make out which gene are you looking for, which is the gene of interest. You will have the size encoded using your knowledge from the genome, and then you can extract that particular part of DNA from the agarose gel. Now, that extraction of the gene of interest from the agarose gel is known as elution. You have to remember this term. So, this is how the DNA is being cut and extracted from the DNA which was given in the first step. The first step that we talked about, the first process, was isolation of the genetic material. Now, once that genetic material has been given, that genetic material has to be cut to give us the gene of interest. And how do we get that gene of interest? For that we have the method of gel electrophoresis, where agarose gel and electric charge is being provided using the nature where DNA is negatively charged. We make the DNA get attracted towards the positive charge. That enables the arrangement in such a way that larger size DNA stays back and the smaller size moves forward. This enables the DNA fragments getting arranged according to their size. When they are arranged according to their size, they are provided with ethidium bromide that makes them glow or as orange bands in UV light. Then they can be extracted using the process of elution. Now, once they have been extracted, the next step demands us to amplify that gene of interest. What it is, please see the next lesson to know it.